Hello again and welcome back to the channel. In this video we're going to be looking at the limiting factors of photosynthesis and how they affect the rate. So the different subsections of this topic will be a recap of the light dependent and light independent phases of photosynthesis. We'll then look at a list of the main limiting factors of photosynthesis. And then we'll think about how light intensity affects the rate of photosynthesis and why, basically where in the process the effect would be seen. We'll also do the same for carbon dioxide, and then we'll finish off by looking at the effect of temperature and where that is seen. So let's dig into it. So, in the previous lesson on the biochemistry of photosynthesis, we looked at the light-dependent and light-independent phases of the cycle or of photosynthesis. So let's start off with the light-dependent phase. So we'll start off with water here, and at the start of the process, we end up with water being broken up into its component parts. And this process is called the photolysis of water and results in the release of electrons, hydrogen ions, and oxygen. So this process is known as the photolysis of water. So in this or during this process, what then happens is the electrons are excited to a higher energy level, and then they end up temporarily binding to a series of electron carriers or photosystems. We start off with photosystem 2 and then we'll go on to photosystem 1. As part of the process, as the electrons go from photosystem 2 to photosystem 1, you end up with enough energy being released to phosphorylate ADP to ATP. That's the first of the two substances we require for the next phase. Once this is done, the electrons and the hydrogen ions recombine along with NADP to form reduced NADP. And that is the second substance we need for the next phase. So that's the light-dependent phase of photosynthesis. Now let's have a look at the light-independent phase. We'll start off with RUBP, which is short for ribulose bisphosphate, which then goes to GP, which is short for glycerate phosphate, and then that goes down to TP, which is short for triose phosphate, and then RUBP can be regenerated. This is also known as the Calvin cycle. Okay, so in this process, you also need CO2, which comes in here. And the reason why is because RUBP has 5 carbons and GP has 3 carbons. So we have a shortfall of 1 carbon atom, and that comes from the carbon dioxide. And you need an enzyme called rubisco for this. So where are the two products of the light-dependent phase required? They're required here between GP and TP. So you've got ATP going to ADP, and you've got reduced NADP going to NADP. So those are the two substances we mentioned from the light-dependent phase. And the whole point of this is then to be able to generate larger biological molecules which the plant might need, such as glucose. And from the previous video, you would also know that the other substances involved are also involved in making or acting as intermediates on the way to making other larger molecules. So that's the recap of the light-dependent phase and the light-independent phase. So let's continue on to the next bit of the page. So what are the two products? I've already mentioned that. We've got ATP and you've got reduced NADP. And the three limiting factors of photosynthesis. So there are three limiting factors of photosynthesis, which you would have done at GCSE. So this is really a recap. You've got light intensity, which is the most obvious, because obviously we need light in order for photosynthesis to occur. And then you've also got the concentration of carbon dioxide. And then finally, temperature. Okay, so on to the next page to discuss the effect of light intensity. What is light intensity? Light intensity is a measure of the amount of light available. So the greater the light intensity, the greater the amount of light available for photosynthesis to occur. So let's have a look at what the effect is of light intensity on the concentrations of the different substances we've talked about. So as you can see from the graph, if we dim the lights and thus reduce the amount of light available, we reduce the light intensity, you end up with a drop in concentration of RUBP and TP. But you end up with an accumulation or an increase in the concentration of GP. So let's write that down. So you can see in the graph there, you end up with an increase in the concentration of GP and a decrease in the concentrations of RUBP and TP. So we need to talk about why that would be the case. So if we go back to page 1, remember that the light-dependent phase, the bit of the process which is affected by light, produces ATP and reduced NADP. And those two substances are required to convert GP to TP. That's important. So let's go back here and explain why we end up with an increase in GP but a decrease in RUBP and TP. So let's write that down. Concentration of GP increases because it is no longer converted to TP as less ATP and reduced NADP are available. And then we'll talk about why the concentrations of RUBP and TP decrease. So the concentration of RUBP and TP decreases because they are still being converted. So they're still being converted into the next steps because they aren't affected by the amount of light available. The only step that's affected is GP going to TP. And because the GP can't be converted to TP, it accumulates and builds up and the concentration increases, whereas the other two substances are still being converted all the way up to GP, and as such, they end up running out. Okay, so let's have a look at the effect of carbon dioxide concentration. What step in photosynthesis requires carbon dioxide? So let's go back to our recap. And as you can see, it's the step between RUBP and GP. So what step in photosynthesis requires carbon dioxide? Conversion of RUBP to GP in the Calvin cycle. So remember that RUBP has 5 carbons in it, whereas GP has 3 carbons in it, but they're 2 molecules. As such, there's a shortfall of 1 carbon atom, which is supplied by carbon dioxide. So a graph showing the effect of carbon dioxide on the process is shown in that diagram. And as you can see, in this case, TP and GP drop, whereas RUBP's concentration increases. The reason why is because the only step in the Calvin cycle which is affected by the carbon dioxide concentration is a conversion of RUBP to GP, which therefore means that if we lower the concentration of CO2, there's less CO2 available for RUBP to be converted to GP, and as such it accumulates, whereas the other 2 molecules continue to be converted onto the next step all the way up to RUBP, and as such their concentrations drop. So let's write down a description of the effect. Number 1. So remember what we said, RUBP accumulates because it's no longer being converted to GP. So concentration of RUBP increases as it is not converted to GP. And then the concentrations of GP and TP decrease because they are still converted on to other substances. And so that's the explanation, or that's a description of what we can see in the graph. So let's put in our more detailed explanations of what's happening. So number one, CO2 concentration affects the conversion of RUBP to GP. Therefore, a drop in CO2 concentration causes RUBP to accumulate or build up, whereas the concentrations of GP and TP increase. Oh, sorry, they decrease. It's all getting quite confusing. So they decrease as they are converted. Okay, so that's the effect of carbon dioxide on the process. So TP and GP drop, RUBP increases, and that's because RUBP is being converted to GP when carbon dioxide is present. And so if it becomes absent, you don't have that conversion. And then finally, let's look at the effect of temperature. So why does temperature have little effect on the rate of the light-dependent reaction? That's because there are few or no enzymes involved. Are few enzymes involved. And if there are few enzymes involved, there's nothing to be affected by the temperature. And as such, it doesn't really have much of an effect on the light-dependent reaction. However, it does have a bit of an effect on the light-independent reaction because it affects the enzyme, the main enzyme that's involved, which is rubisco. So temperature affects the enzyme rubisco in the Calvin cycle. So if we were to draw a graph of the effective temperature on the rate of photosynthesis, you'd end up with something that looks a bit like this. Eventually, the rate will drop. Initially, the rate will increase. That's in step 1 in red. And that's because there'll be more collisions between the different molecules. And as such, the reaction will proceed faster. And then you then get to step 2, which is the optimum temperature. And then after that, the rate begins to decrease, and that's because the shape of Rubisco changes and its action changes. So from the previous worksheet, right at the end, there was an exercise you could do in your own time. And in that exercise, it talked about how Rubisco has two possible jobs. It can act as a carboxylase or an oxygenase. The carboxylase is the effect that we want. The oxygenase is detrimental. It causes something called photorespiration, which just wastes energy. And as such, if you end up with a high temperature, you favor the oxygenase activity, which is not very helpful, and you end up with a decline in the rate of photosynthesis. And then eventually the temperature is so high that the plant dies and you end up with a rate of 0. So let's describe and explain that. Temperature affects the action of Rubisco, which is the enzyme involved in the conversion of RUBP to GP. So a very high temperature favors oxygenase activity, which results in a drop in rate. Whereas lower temperatures, within reason, promote the carboxylase activity, which causes an increase in rate. So in essence, temperature affects the rate or the shape of the Rubisco enzyme, which affects its activity. So that is it on this topic. So let's do a quick look back at the start to remind ourselves of what the light dependent and light independent phases look like. Which leads us nicely on to the next part of this video, which is our summary. So the two main products of the light dependent phase, which are required for the light independent phase or the Calvin cycle, are ATP and reduced NADP. The three main limiting factors that you will need to know for the syllabus are light intensity, concentration of carbon dioxide, and temperature, the ambient temperature, the surrounding temperature of the plant. Number 3, we talked about how light intensity affects the rate of photosynthesis because it determines the amount of ATP and reduced NADP available for the Calvin cycle. So if you reduce the light intensity, you'll end up with less of the conversion of GP to TP. And so you end up with an accumulation of GP and a drop in TP and RUBP concentrations. The concentration of carbon dioxide affects the rate of photosynthesis because it is required for the conversion of RUBP to GP. And as a result, if you end up with a drop in carbon dioxide concentration, you don't convert the RUBP into GP. And so you end up with an accumulation of RUBP and a drop in concentration of GP and TP because those two are still being converted on to the next stages right up until you end up with RUBP being made, and then it just doesn't get converted. And so those two, GP and TP, drop, RUBP builds up. And then we finally closed with ambient temperature and how that affects the rate of photosynthesis. And that's because it affects enzymes. And a change in the shape of the key enzyme, rubisco, means that you end up with basically less of the conversion of RUBP to GP, and all of the other processes slow down. So that is basically it on the limiting factors of photosynthesis.